Hello, students. So we are going to be talking about chapter 5, and chapter 5 states the structure and function of large biological molecules. Now I'm going to give you two synonymous words for biological molecules. We can also refer to them as organic molecules and macromolecules. And you will be testing macromolecules in the lab in the following week. So macro comes from the word large. Therefore, we can call them large molecules or macromolecules. Now, when you look at these large macromolecules or biological molecules, we see that all living organisms are made up of these large biological molecules or macromolecules, namely carbohydrates, lipids and fats is one group, proteins and nucleic acids. Macromolecules, they are polymers built from monomers. So we're going to define what's the difference between a polymer and a monomer and their definitions. Let's go to the smallest one, mono. Mono is derived from the word which means one. Mer is unit. So a monomer is one unit. It's a simple unit. It serves as the building blocks of molecules. Poly means many. Mer, unit. So many units that are joined together form a polymer. So many monomers put together form a large molecule called a polymer. Examples of polymers are carbohydrates, proteins, and nucleic acids. They are all polymers. We've seen here lipids and fats are not considered as this polymer. So here we have a polymer, a very convoluted molecule. We'll be looking at this molecule. This is a protein molecule, but it has a very complex structure. And so when we look at the different structural proteins, I want you to take note of this structural protein that we're going to see because of the alpha helices and the beta pleated structure. So I'm not going to give you the name right now. So we're looking at the synthesis and the breakdown of polymers. How would these large molecules, namely polymers, be broken down into the simpler units called monomers? We have substances called enzymes. So what are enzymes? They are macromolecules that are they are chemical substances. They are specialized macromolecules that are going to speed up a chemical reaction. So either by making a chemical bond or breaking a chemical bond, or making a polymer or breaking down a polymer. So we have two processes, one called dehydration reaction and one is hydrolysis. Dehydration, D means removal. Hydration comes from the word water, hydrate. So when water molecule is removed. So when two monomers, they bond together with the loss of a water molecule, that process is called dehydration synthesis. So you're joining two simple molecules to make a large molecule. So my clue sentence is when you go from small to big. What is the small that I'm considering? Is two monomers. You join two monomers together when they're bonded together to form a large molecule. For example, if you look at a protein is a large molecule, so it's a polymer. What is the protein composed of? It's composed of 20 different amino acids. So Amino acids are the monomers. When you join an amino acid together, it forms the polymer, which is the macromolecule called protein, through dehydration synthesis. Let's look at polymers. Polymers are the large molecules. So if we looked at the protein, now we want to break down the protein into smaller ones. So proteins are broken down to monomers by hydrolysis. And what is hydrolysis? It is the process or the reaction where a large molecule will be broken down to a small molecule with water being added. If water was lost here, water is now going to be added. So remember one reaction. If you remember dehydration, you're taking small molecules, you're taking two monomers, you're joining them together with the loss of a water molecule to make a polymer. Then that process is called dehydration synthesis. Opposite of dehydration synthesis is hydrolysis. Hydro comes from the word water. Lysis is splitting. So you're adding a water molecule to break down the polymer into small ones. So it's the process where the polymer, the large molecule, is broken down. So you're going from large to small with the addition of water molecule. So you take the protein molecule, you break down the protein by adding water, and your end product would be your amino acids. So let's look at the chemical reaction of dehydration synthesis. You have in dehydration reacting, you're synthesizing, you're making a polymer. So you take the small units, short polymers, the short monomers actually, and you are removing water, hydroxyl from this monomer to hydrogen of this, you remove water to make the polymer. So you're taking monomers, you're joining them together to form the large polymer. Opposite is hydrolysis. In hydrolysis, you break down this polymer by adding water to give you your monomers, smaller molecules. You go from large to small. You're going from small, you're joining with the removal of water. So carbohydrates serve as fuel and building material. When you look at the term carbohydrate, I'm going to analyze the term for you. Carbo comes from the element carbon. Hydrate, we just saw, comes from the term, the word water. So what are carbohydrates? Carbohydrate is, the way I define a carbohydrate, it is the hydrate of carbon containing the elements carbon, hydrate is water, hydrogen and oxygen, with water in the ratio of 2:1. So always 2 atoms of hydrogen with 1 of oxygen. Now I'm going to give you 4 simple basic summary questions to answer or to compile all the information for your macromolecule. So the first thing we need to know is the definition. What is the carbohydrate? And I define for you the carbohydrate. It is the hydrate of carbon containing the elements carbon, hydrogen, and oxygen, but with water in the ratio of 2:1. 2 atoms of hydrogen with 1 of oxygen. My next summary question that I want to give you is the structure of the carbohydrate, the types, if any, if that macromolecule has types, and the last point is the function. So let's look at the structure. What is the structure of carbohydrates? We've seen the formula would be CH2O. It is the hydrate of carbon into parentheses here for water to the power of N. So it's the hydrate of carbon containing the elements carbon, hydrogen, and oxygen. And we see now, either you can call it the structure, the form. How do you know you are consuming a carbohydrate? Because carbohydrates exist in two forms. What are they? They occur as a sugar or a starch. So the moment you eat any food items that contain sugars or starch, immediately you know you are consuming a carbohydrate. So carbohydrate macromolecules, they are polysaccharides. We'll talk about the third summary question of mine, namely, they are of three types. What are the three types? Monosaccharides, disaccharides, polysaccharides. Mono comes from the word one. Saccharide comes from the Greek word saccharin, SACCHARIN, which means one sugar unit. So a monosaccharide is a carbohydrate that contains one sugar unit. Now, what are the common monosaccharides? Glucose. The molecular formula for glucose, you must know, is C6H12O6. We could form this here. If this was into parentheses, water was close brackets to the power of N and 6 carbon. So C6, 6 times 2 is 12, 6 ones are 6. That's how you got C6H12O6, which is the most common monosaccharide, glucose. The next common one is fructose. You'll see fructose later. Fructose, into parentheses, fruit sugar. Why are fruits sweet by themselves? It's because of the presence of fructose sugar, which is a monosaccharide. We will be testing these monosaccharides in the lab. The next monosaccharide is glucose, fructose, and galactose. Now, how do we classify these types of carbohydrates? Monosaccharide would be classified by the location of the carbonyl CO functional group, which is an aldose or a ketose. Remember, we talked of aldones and ketones. So aldoses and ketoses. I want to bring to your attention, the moment you see the suffix OSE, immediately you should know it is referring to a sugar. The suffixes of a sugar is OSE, glucose, fructose, galactose, lactose, maltose. These are all sugars. And the number of the carbons in the carbon skeleton would also help to classify whether it's a monosaccharide or a disaccharide or a poly. So we see the aldoses, the presence of the aldoses or the ketoses here. Aldoses, aldehyde group, ketoses, ketone groups. So aldoses, you'll see carbonyl group, CO. So when you see these carbonyl CO groups, will be the aldoses. But in the aldehyde group, you'll see CHO. CHO stands for the aldehyde group. Now, students, I know you're supposed to learn all the structural formulas. I will not ask you to learn all these structural formulas, but you must know at least the functional groups. Now, the difference between what are the trioses? Tri comes from the word three. So there are 3 carbon sugars, C3H6O3. 5 carbon sugars would be the pentoses, hexoses, 6 carbon sugars. So glucose, galactose, fructose, these are all what I just gave you were the monosaccharides. Now, ribose, it's important to remember this term when we talk RNA. It contains, RNA stands for ribonucleic acid because it contains ribose sugar. So take note of these terms here. So we are just looking to see how C6H12O6, which is glucose, is in a linear structure. You can also have ring structures. That's all the take home from here. Now, what the second type of structural carbohydrate is a disaccharide. So di comes from the word two. A disaccharide is composed of two sugar units that are joined together. But what is the name of the bond that joins the two monosaccharides? This is extremely important, and therefore I've highlighted it and bolded it. It's called the glycosidic bond. So this covalent bond is the glycosidic linkage. So I don't just want you to say covalent bond. The most specific answer is a glycosidic bond that joins the sugars in a carbohydrate together, glycosidic linkages. So examples of the disaccharide that we are going to write down, and it would be formed when dehydration reaction is joining the two monosaccharides together. Examples of the disaccharide, which I've come to the next slide here, you will see is maltose. How is maltose formed? When you take two monosaccharides, glucose plus glucose forms maltose. Now, the next disaccharide is sucrose. Very important. Sucrose is table sugar. You are not consuming glucose. We are eating table sugar, which is sucrose. And how is sucrose formed? When you join the two monosaccharides, glucose plus fructose, through dehydration synthesis. So we know sucrose, we know maltose. What is maltose? Malt sugar, two glucose units joined together. And lactose sugar. Lactose is milk sugar. That's why milk by itself has a sweet taste. How is lactose made? When you take glucose and join it with galactose, the other monosaccharide, so lactose sugar is formed. Glucose plus galactose gives you lactose. And lactose, maltose, and sucrose, these are all disaccharides. And then the third structural type of a carbohydrate is poly. Poly comes from the word many. So polysaccharides, there are many sugar units, many monomers that are joined together again by the glycosidic linkages or glycosidic bonds. The polymers they are the polymers of sugars, and they play vital roles in storage and structure, giving structure. So, classic example of a polysaccharide is starch. Where is starch present? Bread, pasta, all these will contain starch. Starch is a storage polysaccharide of plants, and it consists of glucose monomers. Plants, we're going to see, are going to store the starch as granules within the chloroplast and other plastids. And the simplest form of starch is amylose. Amylose, again, is a sugar, which is the simplest part of starch. So we can see here, you have potatoes also contain starch. So they are the storage structures, the plastids that contain storage, that contain the starch granules in the potato cells, contain the amylose, which is the unbranched sugar here. But you see, they're very complex structures. You see the plant cells surrounded by the cell wall. You will also see starch in the cell wall of plants is made up of cellulose. Cellulose is a polysaccharide. So I want you to take note of this. Cell walls in plant. What is plant cell wall composed of? Cellulose. Cellulose is a polysaccharide that is present in plants. Sugar is stored in animal cells as glycogen. So animal cells contain glycogen, which is a polysaccharide. Cellulose is a plant polysaccharide. Animal polysaccharide is glycogen. Where is glycogen also stored? In the muscle cells and in our liver cells. So these are important names of polysaccharides that are exclusively present either in plants or animals. And I've written this here about cellulose. Now, hydrolysis of glycogen in these cells is going to release glucose when the cell requires it, when muscle cells and liver cells require it. Now, there are two different types of glucose. Two forms of glucose is the alpha and the beta glucose. So we are going to see now in the structures, which one can we break down? Now, when we eat the stalk of celery, or you're eating broccoli, or you're eating any hardy vegetables, or you're eating lettuce, or asparagus, what happens? You keep biting and chewing. Classic example, asparagus, and you are left with the tendrils in the mouth. Why? Because humans do not have an enzyme. We lack the enzyme. So we have alpha amylase, we have beta, but we lack alpha amylase to break down the starch. And therefore, all those tendrils remain in the mouth. That's the undigested material. So I'm going to explain it when we come to the slide here. You see, when you look at starch molecule, alpha configuration is largely helical. Cellulose molecule, cellulose, the cell wall of plants, has the beta configuration, and they are straight and they are unbranched. Now, enzymes that digest starch by hydrolyzing the alpha linkages can't hydrolyze the beta linkages in cellulose. So we have the enzyme, the alpha amylase enzyme, to break down starch, but we don't have the beta linkage to break down the starch. And that is why all those tendrils, however much we try to chew celery or broccoli or the stem or the stalk or asparagus, the tendrils, that undigested material will be left in the mouth. The cellulose in human food passes through the digestive tract as insoluble fiber. But why are we eating these plant materials? Because we need to digest. We can digest and get absorbed the soluble fiber, but we cannot digest the insoluble fiber, which is eliminated in our excreta. And some microbes, some microorganisms use these enzymes to digest the cellulose. Many of the herbivores, right from cows to termites, they have a symbiotic relationship. That means they help the microbes to digest, and the microbes live off their gut, all the material in their gut, and help them to digest the food. Now, another type of structural polysaccharide is chitin or chitin. It is found on the exoskeleton of arthropods. Arthropods are your crabs, your insects, etc., spiders. They have the hard shells, and they, excuse me, the hard shell of these insects consists of chitin. Chitin is also going to provide structural support for the cell walls of fungi. Remember, fungi are not plants. Now, lipids are the one class of large biological molecules that does not include the polymers. We saw lipids and fats. Why? Because lipids, they mostly contain hydrocarbon regions and they mix very poorly. If you take water and you mix it to a lipid or a fat, The fat does not dissolve in water. It will stay on the top. It will form oil droplets. It is insoluble in water. So lipids and fats are insoluble. Fats are made up of, how do we define a lipid or fat? A fat, as we saw earlier, consists of 3 fatty acid chains with 1 glycerol molecule. And that glycerol, the moment you hear the suffix OL or the ending of that word is OL, immediately associate it with an alcohol group. So it's a 3-carbon alcohol with a hydroxyl group that is attached to the carbon atom. And we see a fatty acid will have a carboxyl group, COOH group, which is that acid group, to the carbon skeleton. So fats are going to separate from water. Why? Because they are insoluble in water. In a fat, we see 3 fatty acids joined to a glycerol molecule. Therefore, such a molecule is also referred to as triglyceride or triacylglycerol. And these fatty acids in the fat molecule, they can be on the same side or of 2 or 3 different fats. So we've seen 3 fatty acid chains with 1 whole glycerol molecule. This is a fatty acid chain. In this case, it's palmitic acid. So one of the 3 dehydration reactions in the synthesis of the fat molecule. But how are they joined together? In a lipid or fat, there are ester linkages. We saw glycosidic linkages in carbohydrates. In lipids and fats, ester linkages. So you've seen the ester linkages here, the COO group over here. So we see that fatty acids, now the second question, fatty acids are of 2 types, a saturated fatty acid, unsaturated fatty acids. What is a saturated fatty acid? It's a fatty acid that consists of a number of hydrogen atoms that have no double bonds between them. So there are no double bonds between the hydrogen atoms. Unsaturated fatty acids, they have 1 or more double bonds between the carbon atoms. So they have 1 or more double bonds between the carbon and hydrogens, but here there are no double bonds in the case of saturated fatty acids. Second point of a saturated fatty acid, it is a solid at room temperature. No double bonds between the carbon atoms and a large number of hydrogen atoms attached to it. They are solids at room temperature. And the third point is all animal fats will be considered to be saturated fatty acids and they are unhealthy. Unsaturated fatty acids have 1 or more double bonds, and when they have 1 or more double bonds between the carbon atoms, they have lesser number of hydrogen atoms. They are easy to digest. Why? Because they are liquids at room temperature. So your oils, classic example is your olive oil. You can have coconut oil, olive oil, sesame oil, but olive oil is the healthiest. Now here I said animal fats like butter, lard, they are considered saturated. And so it could lead to arteriosclerosis or atherosclerosis, meaning thickening of the arteries, cardiovascular diseases, heart diseases, if you consume a lot of saturated fats. So they are unhealthy. So most animal fats are saturated, may contribute to cardiovascular diseases through plaque deposits. Okay, plant and fish oils are healthy oils. I've just put butter, lard, etc., olive oil. So we talk about hydrogenation. What is hydrogenation? It comes from the word hydrogen bonds. Now we don't want too many hydrogen bonds too, because remember, although hydrogen bonds are weak bonds, but if you have, like in saturated fats, you have tremendous number, maximum number of hydrogen bonds. So they're difficult to break down because you're converting now Hydrogenation is the process of converting your unsaturated fatty acids into fatty acids because you're keeping on adding hydrogen atoms. Hydrogenated vegetable oils will also create the unsaturated fats with trans double bonds. And these trans fats contribute more than saturated fats, so they're very unhealthy, leading to cardiovascular disease. Now, the reason why we eat even carbohydrates or we're eating lipids and fat, because they provide us with energy. Humans and other mammals, we store long-term food reserves in our adipose cells, which are our fat cells. And also, why do we need fat? All the organs in our body, they are cushioned with fat because fat insulates the body and it cushions the vital organs. Now, what are the types of, there are three classes of lipids and fats. One, phospholipids, steroids, two, and the third is, I like to put in the word oils, fats, and waxes. So we're looking at each one. So let's look at phospholipids. Now, what does a phospholipid consist of? It's a group of lipids and fats. So phospho comes from the word phosphorus. And when you look at lipids and fat, what then would be the elements present in lipids and fats? If carbohydrate had CHO group, carbon, hydrogen, oxygen. Remember I gave you the word CHOM? So lipid and fat will have carbon, hydrogen, oxygen, CHO, and phosphorus. P because of the phosphate group. So CHO, nitrogen and phosphorus. Okay, so we have CHOP. So when you look at phospholipid, what does a phospholipid molecule consist of? Lipid have 3 fatty acids, 1 glycerol. So instead of third fatty acid, you'll have 2 fatty acids, the glycerol molecule, which is attached to a phosphate group. Now, the 2 fatty acid tails are hydrophobic. We learn the word hydrophobic and hydrophilic, meaning they fear water. But the phosphate group will form the hydrophilic head. And we see, where do we talk about phospholipids? In the previous chapter, I told you in the previous 2 chapters that phospholipids are an important part of our cell membranes, important component of our cell membranes. A cell membrane consists of a phospholipid bilayer, 2 layers of phospholipids. So what then are the steroids? Steroids are lipids that have a carbon skeleton, but they are consisting of 4 rings that are fused together, joined together. A steroid is a hormone. They are chemical messengers that are released in the body. A very good steroid that our cell membranes are going to make is called cholesterol. So cholesterol is the steroid that is present within our cell membranes. And it is a precursor from which other steroids are going to be synthesized. Now, however, if the cholesterol in the blood increases, it can contribute to cardiovascular diseases. So let's look at the difference now. For a phospholipid, you have 2 fatty acid chains, you have a glycerol molecule, and you have this phosphate group that is attached to it. So instead of the third fatty acid, you have a phosphate group. This is our cell membrane. It consists of 2 layers, a head and a tail. The head is a hydrophilic head, and the tail is hydrophobic because the head faces the watery environment. The tail fears water, water fearing, we are facing inward. So don't forget the phospholipid bilayer is present in our cell membrane. And we've looked at this molecule before. So, which is the steroid. And this molecule would be cholesterol. Okay, this is cholesterol molecule. So I also want to give you that there are two types. When you look at fats, we want to know of HDL and LDL. HDL comes from the word high density lipoprotein. HDL. Go to the doctor, they're talking about HDLs and LDLs. LDL, opposite of HDL, is low density lipoprotein. Now, always think something high should not be good, but in this case, high density lipoprotein is the good cholesterol. So your HDL numbers should always be larger than your LDL numbers. If the HDL number is larger than your low density lipoproteins, you will not suffer from cholesterol. That's what doctors will tell you. Good cholesterol, bad cholesterol. It's your HDL number should always be greater than your LDLs. But if the opposite happens, your LDL numbers are larger than HDLs, then you will suffer from cholesterol. So keep that in mind. Now we come to the third group of macromolecules, namely proteins. So the elements in protein would be carbon, hydrogen, oxygen, nitrogen, the CHON group. Okay, so proteins are accounting for more than 50% of the dry mass of cells, and they consist. What is a protein made up of? A protein consists of 20 different amino acids that are joined together by polypeptide bonds. Now, what are the functions? In this case, we're going to learn a number of functions. So far, carbohydrate, lipids, fats, energy storage molecules, right? They provide us with energy and they store the energy in our cells. But proteins are performing vital functions in our bodies. So proteins, the first thing a protein does, it acts as an enzyme, and enzymes act as catalysts to speed up the reaction. Enzymes are proteins that act as catalysts to speed up a chemical reaction. Other functions proteins are performing would be defense, storage, transport, cell-to-cell communication, movement, and give structural support to the cells. So let's look at each function the protein is performing. So in this case, enzymatic proteins. Proteins are acting as enzymes, and enzymes are going to accelerate or speed up the reaction. So this structure here is an enzyme. And for example, digestive enzymes, they catalyze the hydrolysis of bonds in food molecules. So here, these structures are substrates. And you know, what happens is, you see, the shape of the substrate molecule has to be the same as that of the enzyme in order to fit. So when a substrate goes and binds to an enzyme, it forms what we call the enzyme substrate complex. So if the substrate does not have the same shape of that enzyme, it cannot bind to that enzyme and carry out the reaction. Let's go to storage proteins. Proteins help in storage. Function, storage of amino acids. Example is casein. Casein is the particular enzyme present in milk. It's the protein of milk. It is the major source of amino acids for baby mammals, and plants have the storage proteins also present in seeds. That's why some individuals eat sunflower seeds, pumpkin seeds, etc. Now, ove albumin. Albumin is the white of the egg. Ove albumin is the protein of egg white, and it is used as an amino acid source for the developing embryo. Let's look at hormones. Proteins also function as hormones. They act as hormones in the coordination of an organism's activity. Classic example, insulin. Insulin is the hormone that is secreted by our pancreas. It causes other tissues to take up the glucose, thus thereby regulating the blood sugar level or the concentration of sugar in the blood. So you have insulin being secreted by the pancreas in order to maintain the blood sugar level. Then you have structural proteins, wherein proteins are helping to, you can see this, to contract the muscles. When you contract, you're building your biceps. You relax, you're building your triceps. So they help in the movement. Motor proteins. Motor comes from the term movement. So motor proteins are responsible for the undulations. Undulations are this wave-like motion for cilia and flagella. Now, cilia are small hair-like structures present in protists. You will look at protists when you do prokaryotes and eukaryotic labs under the microscope. So cilia are small hair-like structures present on the outside surface of the paramecium. Flagella. We humans, only the males, the sperm have flagella to swim and meet the egg and unite and form the zygote. But flagella is also present in another microscopic protist called Euglena. So, why can I contract my muscles? Is because of actin protein. Actin protein helps in muscle contraction. Myosin protein helps in muscle relaxation, building of your triceps. Let's go next to defensive protein, another function. Defensive is to build up our immune system, to protect us against diseases. So some proteins are defensive proteins. Example is the antibodies inactivate and help to destroy the viruses and bacteria. We can see with this COVID-19 virus. Now what happens? This is a bacterial cell. Look at the shape of the antibodies. So when a virus or a bacteria tries to attack our cells, our immune system immediately reacts by producing antibodies to fight against the antigen that the microorganism is going to carry with it. Now, in this case, look at the antibodies. The antibody is Y-shaped. Look at the virus. Look at the shape of the virus and look at our antibodies. Now, I want you to remember this one simple statement. Shape of a molecule determines its function. So, if the shape of the antibody is not the same as the antigen, then that antigen cannot go and bind to the antibody and form what we call the antigen-antibody complex, thereby not able to cause the infection. So antibodies can inactivate and help to destroy the viruses and bacteria. Let's look at transport proteins. From the very word, they are moving substances in the body. So transport substances, immediately you should associate it with hemoglobin. Hemoglobin is a transport protein, and I showed you that structural protein right in the beginning, and I said I'm going to talk about it. That was hemoglobin. It was a quaternary structural protein. Very convoluted, very complex. And hemoglobin, how is hemoglobin formed? Hemo comes from the word heme, which is a red iron pigment that attaches to globin, which is a round protein. When it comes in contact with oxygen in the air, forms hemoglobin protein. And so hemoglobin protein is going to transport the oxygen from the lungs to the different parts of the body. And other proteins also are going to transport the molecules across our cell membrane. You can see the transport protein going across our lipid bilayer. Second last protein, receptors. Receptor comes from the word to receive. So receptor proteins are responsible for the cell to receive chemical stimuli, to receive a stimulus and thereby respond to it. So receptors, you can see here, we have signaling molecules and we have receptor proteins that are present on the skin, and the receptors built into our cell membranes. So the receptors built into the membrane of a nerve cell is going to detect that there is a signaling molecule released by other nerve cells. Structural proteins, classic example. So structural proteins give structure to our cell. Why is my hair very thick? Is because of keratin protein. So keratin protein builds up our hair, our nails, the feathers in the birds, the hoofs in animals, the nails in birds, the beaks in birds. Also, insects and spiders, they use silk fibers to make their cocoons and webs. Collagen and elastin are also structural proteins that provide the fibrous framework in animal connective tissue. So you can see collagen protein here. So I told you, functions of proteins are extremely important because we've learned different names of the proteins and the functions they are all performing. Okay. Let's go to the next. We looked at what are amino acids, how they are built together. But when you looked at the amino acid, the polypeptide has a sequence of amino acid. You take note of the carboxyl end, which is called the C terminus, CC carboxyl, and the amino end NH2 will be the N end. Let's go to, and so we see the amino group NH2. So this will be the N end and C end, which is the carboxyl group here, the side chain. These are names of amino acids. When we talk of transcription, translate towards the, for the final exam, the last chapters, we will be building up different proteins. So these are names of some of the proteins. I don't want you to get overwhelmed and learn, although you are expected to know the structures. I am just asking you to be familiar with the names. Don't learn the structures there. Okay, just look at the amino end, and the carboxyl and COH, C terminus end. Now, there are 4 different types of structural proteins: primary, secondary, tertiary, and quaternary. I always say, if the name is difficult to pronounce, remember that's the most complex. So what is primary? I'm just going to give you the basic definition. Primary structural consists of a linear comes from the word line, a linear sequence of amino acids joined together by polypeptide bonds. Secondary, little more complex. So we're building from simple to complex. Secondary structural will consist of coils and folds. Tertiary will have a number of side chains, R groups. So you will have alpha helix and beta pleated structures, because secondary has the helix, alpha helix, beta, and tertiary will be a combination of alpha helices and beta pleated, if you had pleats, folds. Quaternary is the most complex structure with a lot of alpha helices and beta pleated structures. But remember, when you think of quaternary structural protein, immediately associate the example hemoglobin protein. That's all you need to know there. And so they're just showing you a protein from flu virus and the antibody protein here. This is primary structural linear sequence. If you had to open it up, secondary would be this helix with beta chains, beta pleated structures. Trans-theriodine polypeptide is the tertiary, alpha and a combination of beta pleated, and quaternary, very complex, very folded, very convoluted, will be the quaternary protein, which is hemoglobin 2. Okay, so they're showing you the hemoglobin protein here. See, number of alpha helices, and you have also the beta pleated structure over here. So the bridges or the bonds that hold would be called disulfide bridges between a protein molecule. Now, also with the same token, we connect it with the disease called sickle cell anemia. Sickle cell anemia is, or sickle cell disease is, an inherited blood disorder which results from the substitution of one particular amino acid in the protein hemoglobin. And on account of this one substitution, valine with glutamic acid, what happens is these individuals' red blood cells are not normal disc shaped or spherical, they are sickle shaped. And as a result of this, these individuals, they have to go through blood transfusion, etc. They are fatigued, they are in pain, and we'll go into details when I explain to you genetic disorders. So it causes these red blood cells to aggregate into chains in individuals who suffer from sickle cell anemia. Now, what happens to a protein? When can a protein unravel? Unravel means unfold. A protein can unravel when there are alterations in pH, salt concentration, temperature, or even enzyme inhibition. You have these, you'll have the same type of question in your lab when we do enzyme activity. So don't forget that. And this loss of a protein's native structure is called denaturation. So you must understand what is denaturation. And what does denaturation of the protein lead to? It causes the protein to be inactive. Where would you see unfolding, unraveling? You see it in Alzheimer's disease, Parkinson's disease, and mad cow's disease. It's due to the misfolded proteins. The protein should not be folded, misfolded. That is resulting in these diseases. So this was the red blood cell, and this is sickle shaped cell of individual who suffers from sickle cell disease. This is our normal red blood cell, okay? And it is because I told you valine with glutamic acid. So you have valine and glue, which is substituted here. So let's come to the last macromolecule, namely nucleic acid. When you hear the word nucleic acid, nucleic comes from the word nucleus. So they're found in the nucleus of our cells. There are two types of nucleic acids, DNA and RNA. Now, the first thing is I want to give you the differences between DNA and RNA. So DNA, number one, what does the acronym DNA stand for? So the acronym DNA stands for deoxyribonucleic acid. RNA is ribonucleic acid. Number 2 point. DNA is a double-stranded helix. RNA is a single strand. Number 3 point. DNA contains deoxyribose sugar. RNA contains ribose sugar. Then we want to talk about what does DNA have? DNA has thymine present, THYMINE. RNA has uracil present, URACIL. What then is DNA consisting of? On the DNA strand, there are several thousand nucleotides present. A nucleotide is a 3-subunit molecule that consists of a 5-carbon sugar, a nitrogenous base, and a phosphate group. What then is a nucleoside? It has a nitrogenous base and a sugar, no phosphate group. But you don't have nucleosides, you have nucleotides on the DNA strand. Now, there are 2 nitrogenous bases, purines and pyrimidines. What are the purines? Adenine and guanine. There are 6 ring structures that are fused to a 5-membered ring. Pyrimidines are cytosine and thymine. I've written and uracil because uracil is only in RNA, not in DNA. Thymine is substituted by uracil on the RNA strand, as I just told you. So we see here, what are the linkages in nucleic acid? Phosphodiester linkages. You must know it for each and every macromolecule. Now, if this is your DNA molecule, so let's go down to the DNA. This is a double helix. There are 2 strands wound around each other. And on each strand, you have all these 1,000 nucleotides: sugar, phosphate base, sugar, phosphate base, the 3-subunit molecule. Now, these are 2 polynucleotide chains that are forming this double helix. But how does this double helix run? It runs anti-parallel to each other. The backbone is running anti-parallel to each other, with one strand beginning at the 5' position. This is the meaning of prime. And it will terminate at the 3' position. Similarly, the other strand will begin 3' and terminate at the 5'. Now, you have this base pairing taking place, which is adenine, according to what we call Chargaff's rule. Adenine will always pair with thymine, forming a double bond, and cytosine with guanine, forming a triple bond. So you have this molecule called ATCG. That is called Chargaff's rule, CHARGAFFS. So all along here, you'll have a sugar, a phosphate. So that's the backbone, the skeleton, a sugar, a phosphate, and the rungs. You're climbing up the ladder here. These are called the rungs, RUNGS, which are the steps of the ladder, consists of the nitrogenous bases. If you have adenine on one side, it must pair with thymine, forming a double bond. If you have cytosine, it has to pair with guanine, forming a triple bond. So we call it ATCG base pairing. This is the basics of the genetics. Do not forget it. So adenine, which is a purine, will always pair with thymine, a pyrimidine, forming a double bond. Cytosine, a pyrimidine, will always pair with guanine, a purine, forming a triple bond. The skeleton of, or the backbone of the DNA strand are made up of alternating units of sugars and phosphates. And the base, which is the third part of the nucleotide, would be the rungs of the ladder are the bases, nitrogenous bases. So you would have sugar, phosphate base, sugar, phosphate bases, and that forms the DNA helix. And so what is bioinformatics? It uses computer software and other computational tools to deal with the data that results from sequencing a number of genomes. And the way these large sets of genes, or even comparing the whole genome of different species, is called genomics. Now, proteomics is a similar analysis of these large sets of proteins, including their sequences. So, with this, students, there was a lot of information. I hope you will go through this information again. And if you have any questions, feel free to ask me in the lab or attend any of my office hours on Zoom. Take care, students. See you in the lab. Bye-bye.